So a very good afternoon to one and all. I'm working as a bioinformatics scientist at Indian Agriculture Research Statistics, and this is a topic that I very recently have started working on. So this is a very small work that I have recently done. I'll be presenting. So I'd just like to brief about the bioinformatics. So it is basically an integrative approach that integrates statistics and computer applications for analysis of biological data, and it involves innovations in computational technologies to accelerate the research in biology, and specifically we do for crop improvement. So from the last few days we are talking about the plant pathogens, the losses and the economic damage that are done by them. So we are well aware of the fact that they are a huge challenge for us. So in this scenario, the plant pathogen interactions is very crucial for improving crop protection and productivity, and the resistance or susceptibility of plant is basically defined by the molecular exchanges between the plant proteins and the pathogen proteins. Though we have various experimental techniques that can be used for detection of plant pathogen interaction, but they are costly, time consuming and less scalable. So that gives us the alternative to use machine learning and computational methods that can be used for prediction of plant pathogen interaction. Various sorts of omics data that we receive for analysis involves genomics data, transcriptomics, proteomics, epigenomics, metabolomics and phenomics data, and especially after the advent of this next generation sequencing and completion of human genome project, we have huge amount of omics data in our hand that requires advanced computational techniques for analysis. So machine learning gives us an alternative to that. So we are using machine learning to facilitate large-scale plant pathogen interaction identification, because it has already been used for various biological data analysis involving bioimage processing, gene function prediction, protein structure prediction. So that can be used for PPI identification as well. So these terms we come along very frequently, AI, ML and DL. So briefly, AI is a larger domain that encapsulates the machine learning. That AI is basically making machines mimic human behavior, having human intelligence. Machine learning basically identifies patterns from the data to make predictions. Deep learning is further a subdomain that involves neural networks for prediction. So the basic steps that are involved in development of machine learning models starts from data collection, then data processing. Feature extraction is an important step because the data we have is in the form of sequence, and first we have to convert the sequence data into numerical format. Though in deep learning we can directly feed the data and it itself extracts the feature. And then we implement the machine learning algorithms. So based on our problem, whether it is a classification problem or a regression problem. Classification problems is when we have a categorical output. The Y is categorical. Then there can be classes, whether presence of disease or absence of disease, if the proteins are interacting or not interacting. So this is a categorical data. The output is categorical. Then we'll go for classification. If it is a continuous variable, so we'll go for regression problem. That is a regression problem. So in case of quantitative traits, and classification is the case of qualitative traits. If we don't have labeled data, if we just have data for which we do not know what to predict, in that case we go for clustering techniques, in which we cluster together the similar looking data to get some inference out of it. So there are various evaluation measures that are implemented based on whether it is a classification problem or it is a regression problem. If we use accuracy, precision, recall, F1 score, so these are the measures that we use for classification problems. And mean absolute error, mean standard error, RMSE, R square, these are used if there is a regression problem, and these are some of the indices that we use if it is a clustering problem. So when we have the data, first thing is a feature extraction. So what I'll be explaining is basically based on the protein sequence, amino acid sequence data. So the features that we can extract from the amino acid sequence data is amino acid composition, Kremer frequencies. This is basically to convert the sequence data to the numerical data. Then we can extract various physicochemical properties, hydrophobicity, polarity, charge, molecular weight, etc. There are various structural and evolutionary features that can be extracted from the data. Further, if we are not going for feature extractions, we can encode the sequence data into numerical format. So various encodings are available. Most popular ones are the one-hot encoding, bot2vec. So these are the things with which we can convert sequence data to numerical format. So for protein-protein interaction, we can extract basically the physicochemical features, evolutionary features, secondary structures, and these features can be further fed to the machine learning model to train the model and give prediction for the unknown data. So for our study, we have taken the plant pathogen interaction data from these three databases, PHI-base, HPIDB, and PathoPlant. So they have various protein-protein interaction data. So we have taken basically the plant pathogen interaction data, and further we have extracted those sequences from various sequence databases, and we have used sequence-based encoding schemes to convert the sequence data to numerical format. For construction of base model, first we have divided the data to the training data and the testing data. The testing data we'll use to evaluate the accuracy, precision, and all those things of the build model. So first we have taken 90% of the data for the training purpose and 10% of the data for testing purpose. Further for training, we have divided the data into 5 subsets. So this is done for 5-fold cross-validation, so that we utilize whole data for the analysis and testing as well. So in 5-fold cross-validation, what we do is take 4 parts for training and 1 part for testing. So we do it 5 times, so that each of the subset is utilized for training once. So we basically model three of the algorithm based on 3 of the algorithms, popular machine learning algorithm, that is support vector machine, random forest, and ANN. So best performing models that we get out of those models is and we tested on various descriptors, the feature sets that we have extracted. So based on SVM on autocorrelation, random forest on local descriptors, and SVM on conjoint triads. So these are the descriptors. We have got the maximum accuracy, so we have selected these models. Further, to develop the ensemble model, we have given weightage to these models and the probabilities of which we get after the prediction, so to get the final ensemble model. So this is basically the whole outline of the work that we have done. We have taken the data from 3 databases, then we have extracted the features, ran three of the models, and then we have done the ensemble method using the soft voting approach, and then we have got the final ensemble model, and that we tested on other test data that we kept separately before training the data. So after developing the ensemble model, it has achieved the 97% accuracy. That was a bit higher than the individual models. So it shows that the ensemble method is better than the individual approaches. This is one more small computational work that we have done to show how plant pathogen interaction happens. So this does not involve machine learning, but how computationally we look at the plant pathogen interactions. In this we have taken the polygalactonases. So these are the enzymes that are involved in breakdown of cell wall when the infection happens, and the counter protein that is in plant is polygalactonase inhibiting protein that protects the cell wall. It basically starts the immune response and triggers the defense signaling. So to see if there is a mutation in the PGIP, how it affects its interaction with the PG protein. So we have taken the sequence from radish PGIP, and its wilt-causing fungi. So for that we have built the protein structure model, validated that through already available information in other crops. We have identified the active regions that are actively involved in the interaction. So we have mutated those regions, and then we further docked both of the structures before mutation and after mutation. And through MD simulations, we analyzed if the interaction is increased or decreased. So we have seen that after mutation, the interaction between plant and pathogen protein is increased. So it can better help to plant resistance and triggering the defense response in plants. So that could be taken further for wet lab experimentation. So in brief, this is the overall AI and plant defense that can be implemented in various defense research. For resistance gene prediction, we can use it to predict gene expression levels in resistance signaling, predicting the optimal levels of defense related metabolites or proteins, predict stress related genes or proteins, identify potential biomarkers, and predict the efficacy of various different compounds, the secondary metabolites. So the key takeaways from this is we can combine machine learning and omics for studying the plant pathogen interaction, and it can provide a very cost effective, highly scalable technique for proteome wide PPI identification. So I would like to thank fellow scientists, my department, ICR, ISRI, and DBT for the funding. Thank you.
Long live India and France friendship.